In this lecture, we are going to discuss about the basic terminologies involved in terpenes and terpenoids. We will see the definitions, examples, various structures and classification of terpenoids. We will also see how this isoprene rule is useful in understanding the complexity of structures of terpenes and terpenoids. Terpenes and terpenoids are the natural products, and these natural products are mainly synthesized from the plants. These terpenes and terpenoids are made up of the isoprene unit. In other words, this isoprene molecule is found to be a repeating unit in the structures of terpenes and terpenoids. When these compounds contain carbon and hydrogen, it is called as a terpenes. When it contains carbon, hydrogen and oxygen, then it is called as terpenoids. In other words, the oxygenated form of the terpenes is called as terpenoids. In the literature, more than 23,000 terpenes are known. We find various applications of terpenes in our everyday life. Let us see some of the examples. The first example is of myrcene molecule. Myrcene is an acyclic molecule containing 3 double bonds and it contains 10 number of carbon atom. It is a major constituent of bay leaf here, and bay leaf is used as a spices for making Indian curries. Second example is of alpha-phellandrine. Alpha-phellandrine is a monocyclic compound containing 2 double bonds. It's a homoannular diene. It is a constituent of eucalyptus oil, and eucalyptus oil is having some medicinal importance. This is a structure of geraniol which is present in rose oil. Alpha menthol is present in the leaves of peppermint. Alpha terpenol is present in the seeds of cardamom. These three examples contain oxygen in the form of alcohol functional group. It's a primary alcohol, it's a secondary alcohol, it's a tertiary alcohol, and since it contains carbon, hydrogen and oxygen, they will be called as terpenoids. Let us take few more examples of terpenoids. This is a molecule of carbon. It is an alpha beta unsaturated ketone. It is present in caraway seeds, and caraway seeds is a main constituent of spices. This is a structure of gingiberine molecule, and gingiberine contains 15 number of carbon atoms. There are 3 double bonds. It's a homoannular diene, and this gingiberine is a main constituent in ginger. The pleasant smell of lemon is due to presence of a terpene called as citral. Citral is an alpha beta unsaturated aldehyde, and here it again contains 10 number of carbon atoms. These are the structures of alpha pinene and the beta pinene. Both of them are bicyclic structure, and this alpha pinene and beta pinene is having some historical importance. In the ancient days, from African jungle, people were extracting from resins of pine trees, and the distillations of these resins of pine tree leads to a chemical called as turpentine, and turpentine contains alpha pinene and the beta pinene as major constituents. So because of the turpentine oil, the words terpenes and terpenoids are derived. The last molecule is very interesting. It is called as a beta carotene. Beta carotene contains total 11 number of conjugated double bonds, and because of this conjugated chromophore, it is having very high lambda max value, 456 nanometer. The red color of the carrot is due to the presence of beta carotene. So if we look at all these examples of terpenes, we find that they contains carbon atom in multiple of 5 here. Isoprene is a repeating unit, and isoprene contains 5 number of carbon atoms. So myrcene to carbon contains 10 number of carbon atom. Gingiberine contains 15 number of carbon atom. Vitamin A contains 20, squalene contains 30, and beta carotene contains 14 number of carbon atoms. So terpenoids are mainly made up of the isoprene units, and the number of carbon atoms present in them depends upon how many isoprene unit contains. So in other words, terpenes are made up of isoprene units. As far as the structures of terpenes are concerned, it can be the acyclic structure like myrcene molecule, geraniol molecule, citral molecule. It can be the monocyclic, for example, phellandrine, limonene molecule, menthol, alpha terpenol, all these are monocyclic. In bicyclic variety, we may have an example of alpha pinene, beta pinene. Tricyclic structure like santonin is a sesquiterpenoid, and it is having a tricyclic structures. So it can be the terpenoids can have acyclic, monocyclic, bicyclic as well as tricyclic structures. As far as the oxygen functionality in terpenoid is concerned, alcohols, aldehydes and ketones, these are the 3 common functionality. functional groups in terpenoids, whereas acids and esters are also exist in terpenoid family, but acids and ester containing functional groups are very less available in terpenoid family. Now let us see the classifications of terpenoids. As already discussed that terpenes are made up of isoprene units, and depending upon how many isoprene units it contains, the terpenes are classified into mono, di, tri and tetra. Monoterpenes contains 2 number of isoprene unit, di contains 4 number of isoprene unit, tri contains 6, tetra contains 6. That's a 2, 4, 6, 8 number of isoprene unit corresponds to the monoterpenes, diterpenes, triterpene and tetraterpenes. When it contains odd number of isoprene unit, it is called as a sesquiterpenes. Sesqu means 1.5 and sister means 2.5. So one can have a sesquiterpenes. There's a 3 number of isoprene unit. Sister terpenes is a 5 number of isoprene unit. Now corresponding to these isoprene units, we will have the number of carbon atoms in the multiple of 5. C10, C20, C30 and C40 number of carbon atoms are present in monoterpenoid, diterpenoid, triterpenoid and as well as tetraterpenoid. When it contains 15 number and 25 number, it is called as a sesquiterpene as well as sister terpenes. Corresponding to each class of the terpenes, one should have at least one example on our fingertips, like citral menthol for monoterpenes, squalene is for triterpene, tetraterpene examples are lycopene and the beta carotene. Compared to any other class of the terpenoid, the sister terpenoids, the examples of sister terpenoids, that is C25 number of carbon atoms, are relatively less in the literature. The lower class of the terpenoids, mono and sesquiterpenoids, they are relatively steam volatile, contributing to the development of essential oils. When this isoprene contains more than 100 number of when this terpenoids contains more than 100 isoprene units, the resulting molecule may be called as a polyisoprene, and polyisoprene is called as a natural rubber. So this is the classification of terpenoids based upon the isoprene molecule. We have already said that the terpenes are made up of the isoprene molecules. Isoprene molecule is a branch molecule. The branch portion is called as a head and unbranched portion is called as a tail. We can visualize here in the form of this cartoon where this head portion is a part of branch and a tail portion is a unbranched portion. So theoretically there are 3 possibilities that these 2 isoprene units can be joined through head to tail linkage, tail to tail linkage and head to tail linkages. But the nature is very systematic. It is observed that most of the part of the structures of terpenoids contains isoprene units joined through head to tail linkage. So this linkage of isoprene unit through head to tail linkage is called as a isoprene rule. This isoprene rule was proposed by Otto on the basis of 2 important observations. The first observation is that the empirical formula of all terpene was found to be C5H8. And the second observation is that the thermal decomposition of all terpenoids gives several products and one of the product was found to be the isoprene. And since this is a product form, on the basis of these 2 observations, Otto reached to a very important conclusion that terpenes are made up of isoprene units and they are joined through head to tail linkage. And this statement is called as isoprene rule, or it is also called as a special isoprene rule. And this special isoprene rule is useful in understanding the structures of these terpenoid molecules. In a couple of slides, we will see how this is applicable to the class of monoterpenoid, sesquiterpenoid, di, tri and as well as tetraterpenoids. Monoterpenoids contains 2 isoprene units and monoterpenoids can be acyclic, it can be monocyclic, it can be the bicyclic. Citral, geraniol and myrcene, these are the examples of acyclic monoterpenoids, and let us ignore here double bond as well as functional group. It is made up of 10 number of carbon atoms, so this is the one isoprene unit containing 5 number of carbon atom. This is another isoprene unit containing 5 number of carbon atom. Here head is connected to the tail, a branch portion is connected to the tail. Here also a branch portion, head is connected to the unbranched portion, that is a tail. Here is also head is connected to the tail. So in monoterpenoids we find at least one head to tail linkage. Here you do not find head to tail linkage. Here also there is no head to tail linkage. But please note down that when we will discuss the biosynthesis of monoterpenoid, at that time we will realize that in the beginning of enzymatic transformations, first of all head to tail coupling occurs and through other enzymatic transformations this cyclizations occurs. Then in sesquiterpenoid again 3 isoprene units are joined. One can have a acyclic sesquiterpenoid, monocyclic sesquiterpenoid, bicyclic, tricyclic structures also exist. In acyclic terpenoids we are having 2 two head to tail linkages. Here is again two head to tail linkages. So in sesquiterpenite we find at least two head to tail linkages, and here again we find a branch portion connected to the unbranched. The same thing is applicable to the sesquiterpenite molecule. In diterpenate contains 4 isoprene units. This is an example of phytol molecule, which is a substructure unit in chlorophyll molecule. Here the 4 isoprene units are beautifully connected through tail head. Here is a tail head, here is a tail and head. Vitamin A is again a diterpenate where 4 isoprene units are there. Vitamin A is involved in the chemistry of vision of an eye, and here again it's connected to tail to head. Here is a tail to head, tail to head. 3 head to tail linkages are observed in case of diterpenate, that's a vitamin A. Now we say that terpenoids are made up of the isoprene unit and they are joined through head to tail coupling. This kind of the isoprene rule is a very useful rule in understanding the structures of the terpenoid family, but it is not a fixed rule. It can be used only as a guiding principle because certain exception occurs. However, these exceptions are very less in number. For example, levandulol. Levandulol is a monoterpenoid, but here we do not find head to tail coupling. Similar exceptions occurs in higher terpenoids like triterpenide as well as tetraterpenoids. In triterpenide and tetraterpenide, we find head to tail linkage by and large in major portions of the molecule, but at the central portion of the molecule there is a tail to tail coupling. We will see specifically in case of squalene molecule. Squalene molecule is an example of triterpenoid. It's a triterpene, basically squalene molecule, and there are 6 isoprene units. These 6 isoprene units are joined again through head to tail linkage, but at the center portion of the molecule there is a tail to tail linkage, but remaining portion again there is a head to tail linkage. So isoprene rule is violated here, but it's a very again useful one because in rest of the portion of the molecule there is a head to tail linkage. Tetraterpenite contain 8 isoprene molecules like beta carotene and the lycopene. All of them are containing carbon hydrogen, so they can be called as terpenes, and specifically it's a tetraterpene. Now again in large portion of the molecule there is a head to tail coupling in beta carotene, but at the central portion of the molecule a unbranched portion connected to the unbranched, and therefore here is a tail to tail coupling. Lycopene is present in tomatoes. Again here head to tail linkage is observed, but at the central portions of the molecule there is a tail to tail linkage. So in this way we have seen that terpenes are made up of the isoprene units and they are joined through head to tail linkages. Let us see its utility where they are used. It is observed that monoterpenoids and the sesquiterpenoids are steam volatile compounds, higher order, higher range of terpenoids, lower range of the terpenoids, and they are major constituents of the essential oils. Essential means essence, that's the aroma, and because of this aroma, they are commercially available in the market. For example, rose oil, then mint leaves, menthol, alpha phenanthrene, myrcene, alpha terpenol, alpha pinene, beta pinene. These are the constituents of several essential oils sold in the form of, for example, orange oil, palm rose oil, lemongrass oil, citronella oil. These are commercially available and it is having some applications. Some of the applications we find in the food industries, they are used as natural flavor additives. They are used as fragrance in perfumery industry, and they are also having certain medicinal property. For example, the eucalyptus oil is used as a medicines. Then as far as the aroma industries is concerned, we find that 36.80% of terpenes is contributing to the 2016 market share, and therefore the terpenes is having tremendous utility, commercial utility. Now let us see, if suppose the terpenes are not there around us, what will happen to our beautiful world? So first of all, whatever the spices we are using, particularly in India, for making the curry, then this spices contains terpenes as well as terpenoids. And if there is no terpenes, then the question is what we will eat then, and it will again about the taste of the prepared kind of curry. Then these are the essential oils. They are used as perfumes as well as deodorant, and suppose they are not, no perfumes and no deodorants, then definitely Jack will be reluctant to go to the party. Rose will not smell the way it does, and that's why there's a big problem from the youngsters. And lastly, that we have seen the polyisoprene can be used as a rubbers, and if there is no terpenoid, there will be no rubbers, no tires for vehicles, no balloons and no gloves, and therefore we conclude that the terpenes are indeed very essential. So this is the last slide, and we will make a final conclusion about what we have learned in this lecture. From all this structure, it is very clear that terpenes and terpenoids are natural products. They are synthesized from the plants. Naturally occurring compounds synthesized from the plant contain isoprene as a repeating unit. The number of carbon atoms are in the multiple of 5. They contain isoprene, and isoprene is joint head to tail linkage, and as far as their utility is concerned, they are used as essential oils. So this is all about the basic introduction, basic terminologies involved in terpenes and terpenoids. The next lecture will be related to the structuration of citral. I have decided to go for the structure determinations, reaction, synthesis, and stereochemistry whenever it is applicable for the dozens of molecules like citral, geraniol, menthol, alpha terpenol, camphor, farnesol, santonin, gingiberine, phytol, abitic acid, vitamin A, and beta carotene. So we will discuss in detail about the structure determination, reaction, synthesis, and the stereochemistry whenever it is applicable. For example, menthol is having a very rich stereochemical aspect. We will discuss in very detail. We will also discuss the genesis of biological isoprene unit. That's a biosynthesis of terpenoids, how nature prepared this kind of the molecules. Very interesting. Carbocation chemistry is involved in the biosynthesis of terpenoid. It is divided into 2 lectures, part 1, and the second lecture is again related to the actual biosynthesis of terpenoid molecules. And the lecture number 16 is related to the pericyclic reactions of the terpenoids. In some reactions of some terpenoids undergo some sort of pericyclic reactions. We'll discuss it. And the last lecture will be related to the multiple choice questions in terpenes and terpenoids. The purpose of last lecture is a revision kind of the exercise, what we have learned out of all these efforts. So the next lecture is on citral. Thank you very much for watching the video.